The following is a project supported through the Canadian Agri-Safety Applied Science Program, administered by Agravita Canada. Development and assessment of emerging green technologies to reduce aerosol risks and hazards in farms. Airborne contaminants from livestock facilities such as dust, gases, and microbial pathogens can be harmful to both humans and animals. These contaminants also put the health of rural communities at risk. Studies have shown high rates of respiratory illnesses in animal farm workers due to the contaminants present in livestock air. Disease outbreaks within a herd can be caused by the airborne transmission of microbial pathogens and have devastating economic impacts on both farms and the industry. The main goal of this project is to mitigate these hazards by reducing dust and microorganisms in livestock facilities. This will enhance health and safety in agricultural production. To achieve this goal, 2 emerging green technologies will be adapted and improved for application in specific livestock facilities. Electrostatic precipitation, or ESP for short, will be tested to remove dust from poultry houses. An electro-nanospray system will be used to generate engineered water nanostructures and deactivate microorganisms in swine barns. ESP uses electricity to charge airborne dust particles, which can then be collected on a surface with the opposite charge, much like a magnet. The research team tested the ESP technology in both medium and large-scale poultry houses. In both experiments, control rooms were compared to test rooms equipped with an ESP. A corona pipe connected to a high-voltage power supply was used to impart a negative charge to the dust particles. All the metal parts in the test rooms, such as walls or chains, were then grounded and used as collection surfaces. Results showed that the ESP performed well, with removal efficiency up to 62% for total dust, 63% for total bacteria, and above 50% for odors. However, it was observed that the performance decreased as the trials progressed. This could have been caused by the considerable accumulation of dust in the collection surfaces. The ESP didn't seem to affect the health of the broiler chickens used in the experiment, since no significant differences were observed in either the mortality rates or the feed conversion ratios. These experiments demonstrated the potential for ESP to be used on a large scale in poultry houses. However, more work is needed to maintain performance over time and to evaluate the installation and operating costs of such a system. The electro-nanospray technology is used to generate engineered water nanostructures, or EWNS, which contain highly reactive compounds. These compounds react with microorganisms and interfere with their metabolic processes. The objective is to effectively deactivate any harmful microbes. The development of this technology is not as advanced as the ESP and required more testing at the laboratory scale to determine the optimal operating conditions. The EWNS were produced with a lab-scale nanospray system. A syringe pump was used to force a water-based solution through metallic needles connected to a high-voltage power supply. The needles therefore acted as charging electrodes. E. coli was used as a model pathogen for both air and surface decontamination tests. Excellent results were obtained for surfaces with inactivation rates of up to 4 log or 99.99%. Good results were obtained for the deactivation efficiency of airborne bacteria, but the values were much lower than with the surface decontamination tests. The EWNS droplets fell down through the contaminated air as it flowed across the test chamber. Therefore, the contact between the microbes and the EWNS was not as efficient as with the surface tests. Following the lab-scale tests, a pilot-scale nanospray system was developed for use in an actual swine barn at the Prairie Swine Center near Saskatoon. The tests were conducted in a small-scale pig room, which can accommodate 4 grower-finisher pigs. The nanospray system consisted of a unit with 16 needles with an airflow rate equivalent to 10 air exchanges per hour. Preliminary results from the pilot-scale tests are promising, with bioaerosol concentrations reduced by up to 80% through the treatment chamber. From the in-barn tests, the pilot-scale system was able to achieve an overall bioaerosol reduction of 24%. Final results will be available once all the tests are completed. The reduction of airborne contaminants in livestock production is paramount to animal health and productivity, as well as to the health of those who work in these environments. The tests carried out during this project demonstrated the feasibility of adapting emerging green technologies for use in agricultural settings. The ESP installed in poultry houses efficiently reduced dust, microbes, and odors. The nanospray system was able to deactivate potentially pathogenic microorganisms from swine barns. Please visit our website for more information on all the projects of the Canadian Agri-Safety Applied Science Program.